Okay, so in this video, we're going to look at the electron transport chain. We're going to look at chemiosmosis, osmosis, and oxidative phosphorylation via chemiosmosis and the electron transport chain. So again, this is very similar to photosynthesis in terms of electron transport chain, ATP synthase, production, those sorts of things. But kind of the source of electrons is different. So remember up here, we had our net stuff we made in the Krebs cycle. So I'm just going to put that right up here for now, just so we have that. So remember that. So we have this NADH and H plus ions. We have 2 FADH2 and 2 ATP. ATP is fine, it's used. So now it's about what happens here. How do we generate that other approximately 34 ATP molecules that need to be produced here? So this is inside the matrix here, and this is the intermembrane space. Okay, so if you're looking at a mitochondria, this is where we're at in here. Sorry, let me get a right color. So we're in here with the matrix, okay? And then with the intermembrane space, we're out here, okay? This kind of thinner space is out there, okay? So remember, in the matrix, this is where the link reaction occurs, and sorry, not glycolysis, that's the cytoplasm, and the Krebs cycle, or bicycle, because it spins twice with each glucose molecule. But the link of the Krebs reaction occurs in here. So what we have, how we start out this whole process is we have NADH here, right? And remember, NADH is carrying electrons. It's carrying these high energy electrons that were pulled off of this glucose molecule through various processes. So it's right here, we're just taking off a bit of energy there, right? The electron transport chain is where we cash it in. So what happens is NADH drops off electrons, okay? This says 2, sometimes you might see it just as 1. It's not too important. But it drops it off at the electron transport chain, which is kind of this area here, okay? So the electron transport chain. And those electrons go to these various complexes. They're transferred through. Now this is on the inner membrane, okay? So this whole inner membrane here, so all along here, is simply packed with electron transport chains. So it moves through these, and it comes to this one called cytochrome C, and then cytochrome C passes this electron off to hydrogen ions in water, plus those electrons to form H2O, right? Now we call H2O as the final electron acceptor, okay? So at this point, we've stripped all the useful energy off of this electron, so water just takes it. So then what happens though is as it moves through the electron transport chain, just like in photosynthesis, H plus ions are actively transported out, okay? So they're moved out. Now notice for each electron that's dropped off, it can move 3 hydrogen ions through, roughly, okay? So we go from a low H plus concentration to a high H plus concentration, okay? So the movement there creates a high concentration out here, out in the inner membrane space, and a low concentration in the matrix, okay? So now the other one we talked about is FADH2, and I said that's kind of like the scooter while the NADH is a big truck. So FADH2 drops its electrons here, okay? So it can only move through 2 hydrogen ions. So it's got a little bit less energy. So because it drops it here, it only has enough energy to move that one through before it joins with water. Okay? And then that turns into FAD plus, and FAD plus then goes along with NADH and goes back into the Krebs cycle to pick up another electron, and this whole cycle can continue. Okay? So again, that doesn't make any energy yet, but what it's created is a gradient, and we say a proton gradient. If you wrote an H plus gradient on the test, that would be fine. On one side of the membrane and on the other. Okay, this is where chemiosmosis comes in. Remember, osmosis was a movement of water through a semi-permeable membrane. Chemiosmosis is a movement of chemicals through a semi-permeable membrane. In this case, hydrogen, right? They can't go through this way. They can't go through any of the electron transport chains here because they only go one way. The only way it can go through is through this molecule called ATP synthase. Okay? So that's why it's osmosis. The semi-permeable membrane, it only lets those hydrogen ions through. So these are going to want to move from a high ion concentration into the matrix. Now, as one hydrogen ion moves through, you form an ATP molecule. Okay? So for every hydrogen ion that moves through, you form an ATP molecule. Okay? This is the idea of oxidative phosphorylation. Oops, sorry about that. Phosphorylation. Okay? In the sense that the electrons provided the energy and the oxygen is the acceptor. That's the oxidative part. Phosphorylation here is you're adding an inorganic phosphate to ADP to make ATP. All right, so let's do the addition here now. So for every one NADH molecule dropped off on the electron transport chain, 3 ATP molecules are produced. That's because each H plus ion goes through, and remember, NADH can move 1, 2, 3 hydrogen molecules through because the electron travels on there. That electron has enough energy to move those 3 hydrogen ions through. And then those 3 hydrogen ions will go through here once, twice, and a third hydrogen ion will come through. Okay? And each time one of those goes through, you make ATP. So it can make a total of 3 ATP. Now, FADH2 can only make 2 ATP molecules. Okay, so 1 makes 2 because it drops it off here. So 1, the electrons go through here once to cytochrome C and then to here. That provides another energy there. So there's only 2 hydrogen ions that get moved through. Those 2 go back through into the matrix through ATP synthase, producing 2 ATP molecules. All right, so now we can do all this math, right? So if we have 10 NADH molecules, that's equal to 30 ATP molecules. This is equal to 4 ATP molecules. And the 2 we have, we get our 36 ATP molecules, right? And that's how much energy kind of is in a glucose, okay? Again, sometimes you see this is 32, 38. It kind of depends on how you do the math and what you calculate into that in terms of transport costs and things. But for IB land, we'll stick with 36, okay? So hopefully that explains the electron transport chain and that whole process. Now this isn't that much different from the light-dependent reaction. The only difference here is the electrons are coming from water, right? But you're still basically making the same process here. You can see the movement of electrons moves hydrogen ions through, generating all that electricity. The locations are different, obviously, but the process is pretty much the same with the addition of NADPH, P for photosynthesis, rather than NADH. Okay, so I hope these videos help with your studying and your revision, and thank you.